Some, again, interesting insights there, Joel, and really nice to see some of the recent developments being used or being trialled at the border. Now we're going to wrap up our Future of Technology in Action session today with Brendan Rodoni. He's Research Director within Agricultural Victoria, and he's going to talk about the collaborative project to advance the use of high throughput sequencing in biosecurity. It's another great example of strong partnerships in action. Over to you, Brendan.
Thank you very much. And you can see my talk okay?
No, it hasn't come through yet, Brendan.
Oh, good start.
I'll let you know. There, it's coming now.
Thank
Just
you.
need to go to presentation mode. Perfect.
Okay, thank you very much, and thank you to Malcolm and QDAP for giving the opportunity to talk to you about high throughput sequencing with a biosecurity context. So what is high throughput sequencing? It is very much a disruptive technology for diagnostics, and I often say it's the difference between having a landline telephone and our mobile telephones we have now. It's almost comparing from PCR to HTS. So it's a massive change for us, and I think we'll hear more and more about it, and I'll show you a very good example of that in a minute. But the point I want to make here over on the right is the blue line is what was the actual use of HTS in 2018. The red line for each dot is what the anticipated rate is in 2026. And this one here is for diagnostics, and that's human diagnostics. Now, with COVID, I'd imagine that dot is way, way higher. But over here is actual agricultural usage. And I do think too that the rate of uptake and use of HTS in agriculture for breeding, for diagnostics, for surveillance has increased enormously in the last 4 or 5 years. And the other point to make is that high throughput sequencing has many, many different applications. And if someone says to you they're using HTS, they're probably using it differently to the person sitting next to them. So we've got different platforms, we've got different approaches and different applications, and these are really important things. And we will use this. We are using HTS and will use it for genome characterization, identification and taxonomy of pests and pathogens. It's an incredibly powerful tool for pest and pathogen detection, and also is really handy for genomic epidemiology. And so this one science can do characterization, it can do identification of organisms, it can detect them, and it can also be used as an indication. And the great example of the indicator is on our daily news when we hear about a COVID detection in sewage water in some suburban sewage treatment plant or regional treatment plant. So that's an example of using it as an indicator. The thing I think that makes HTS really useful for diagnostics and biosecurity is that it's scalable. And if you look at this standard workflow up the top here, we've basically got our sample and we go through these 4 or 5 steps and we get our data. The basic steps are sample handling, there's nothing new there. DNA extraction, been doing that for 30 years with PCR. The big changes here are around the sequencing and the data analysis. And so this is where a lot of our investment is. But if you look at these steps, a lot of that can be automated. And if it's automated, then we can do large numbers. And when we do large numbers, all of a sudden we reduce our labour costs. And this, to me, for stakeholders, is a key thing in that HTS used the right way in an accurate way will also be affordable. But it does require investment in infrastructure, and that's around the sequencing and computer power. This, like with Joel's talk, we're talking about big data generation here. and we need a lot of computer power to analyse the data sets. And absolutely key from a biosecurity point of view is that trusted databases. And the most obvious example I can give to you of how society is using HGS now is with COVID-19 and contact tracing. We hear about this daily, where we have sick persons get detected with the virus, and we sequence the virus, and then we use a database. And these last two steps essentially is high throughput sequencing. And a really important part of this is this national database and facility that enables our jurisdictional labs to share data and compare and quickly get a result to our stakeholders. In this case, CHO of Queensland, and down the bottom here, it enables people to track the movement of a pest or pathogen, in this case, the virus. So that to me is the most obvious example of how HGS is being used in our daily lives. Did want to put in a quick context around biosecurity, and then come back to how we use HGS in a biosecurity format. So these stats you've probably heard already this week, but we import, when we're not in a pandemic, we have very active airports with people entering. We have an enormous number of sea cargo consignments coming to the country, and just heard about the mail centres. And the big thing there, whatever that number is, we expect this to increase significantly. So this slide was generated by DORA in 2019, and they predicted a 100% increase between 2019 and 2025 in containerised cargo. And so we can only expect the pressure is going to increase. And say in animal biosecurity, some of the pests or the pathogens that we are at risk from could have a significant impact. And the important thing is these percentages in figures, that's what we predict is the likelihood that we'll have an incursion of one of these pathogens in the next 5 years. So for African swine fever, with it on our northern borders now, there's 21% chance that Australia will experience an incursion of African swine fever, and so too with African horse sickness, foot and mouth disease, and lumpy skin. So the significance of these pathogens are enormous. And when we look in plant biosecurity, the number of incursions are increasing annually. And so, if we look at, we're seeing an increase in interceptions of biosecurity risk materials at our borders by almost 50% to 337,000 between 2012 and 2017. So we're seeing a massive accumulative burden on interceptions, and for each of these cases, at some point there is diagnostics. And this slide really emulates that for me. And so in plant biosecurity, we've got a national priority plant pest list. There's the list of 42, which many of you have probably heard about. In the last 12 months, we've had interceptions of 10 of those 42, where not so much interceptions, but where we've had situations where we need to do a diagnosis, or we need to look more deeply at is this organism present. So just in those top 42, the enormous pressure in the last 12 months is quite significant. So how can we use high throughput sequencing to help us with that pressure? We need to get smarter and quicker. And the example I want to give is actually a Queensland example in collaboration with our lab in Victoria, with Beck Roach, who was a PhD student at Eco Sciences, but now works for QDAF. And Beck had a PhD on Xanthomonas species in Chile, and she used high throughput sequencing for genome characterization and identification and taxonomy. She did this beautiful tree over here with the different colours representing different species, and she found out a few really interesting things. So with this species here, not present in Australia, so now we know it is a biosecurity risk. she actually, in the green below, Xanthomonas species, she actually detected some new organisms we didn't even know exist globally, and so they need to be characterised. And with the pink species, X perforans, we knew that it only caused this disease in tomato and not chili. So really valuable, powerful information for our breeding programs when trying to breed for resistance. So that's a nice example of how HTS is used for genome characterisation. For surveillance, there's some really nice work going on around metabarcoding and where we do an amplification and sequencing amplicons. And this metabarcoding can be used for eDNA purposes as well. But the example we've been working on in Victoria is around trying to, over on the left, trying to see if we can use metabarcoding and extracting the DNA from that mixture of insects and identifying individual species. And it actually works quite well. And our theory is that we can do those 5 steps. So we sample our insects, we extract the DNA, we do a PCR, we sequence, we do our data analysis, and we can identify multiple species in the one reaction. And it is absolutely feasible for us in the one sample to detect TPP, Russian wheat aphid, onion thrips, brown marmorated stink bug, spotted wing Drosophila, capra beetle. We can detect hundreds of samples to species level in the one test reaction. From that scalable, back to that HTS scalable point of view, we've actually done an economic analysis of what time it takes for us to do that. And for us to process our insect traps or collection of insects in a semi-high throughput level, we actually can generate this analysis for around about $150 per pot, and that's probably only twice as expensive as what our routine PCR high throughput costs are. So, and we think it'll get a lot cheaper in that 28% of our labour was actually getting the insect into the tube to do the DNA extraction, and we know we can improve that step. So I really think, if we can analyse a population of insects to species level for around about that $100 mark, I think all of a sudden this technology will become affordable for all sorts of surveillance programs. And just that background investment around the databases to support this metabarcoding for insect detection. We've actually now got a database of 200,000 trusted gene sequences of different insect species that we compare our environmental sequence to make that identification, and it's growing every year. And that's the beauty. That's the beauty of our databases. We can build on them, just like the algorithms Joel talked about. We can build on those and make our databases more impactful, more reliable, and also more localised at an Australian or state level. So metabarcoding is, I think, it could be a game changer for surveillance and collecting data. The third one is very similar to the AUSTRAC. So this work is a pest genomic epidemiology program that we worked on, and this is a set of data that was done on Queensland fruit fly, and it was done by Alex Piper and Paul Cunningham in the AgVic lab. But essentially, we've got in the middle, we've got our sequencer. So we sequenced genome skin populations of Q fly, and from our analysis, we'll be able to look for recombination events and exchange of DNA in that eukaryotic genome. But we're also able to type the species into like groups, and then if we overlay that typing with geographic location, we can all of a sudden start to map populations of Q fly and how they've moved. And so this genomic epidemiology took Alex a long time to do, but we will get much quicker at that, and we absolutely believe we can use it live in a disease incursion. Just hot off the mail, I guess, even though it took us 2 months, we Also, we're able to use this genomic epidemiology to map and attempt to trace the movement of the avian influenza virus outbreak that we had in Victoria last year. And we now have really strong evidence that wind was the dispersal agent between one infected property, IP1 and IP3. So that sort of information is incredibly powerful in terms of getting decisions for our regulators. So biosecurity and HGS, a really key thing. We've got our big diseases, we've got our pipelines in the middle. The issue is that we have to get the analysis right. We have to do our tests at a standard that is acceptable and accurate. We have to minimise the opportunity to make mistakes. And it's, another analogy is we can have good car drivers and we can have bad car drivers. And we want to do HDS at that good level all the time. So we see this, all of us feel this in biosecurity, where we're a part of an international and national and state guidelines and legislation. So from OIE or IPPC to our national IGAB agreements that our states and federal government sign up to, all the way down to the legislation in our states, we need to operate to a standard within these frameworks. And that goes for diagnostics as well. And so, as Malcolm mentioned at the start of this session, the NBC did endorse a working group to develop a framework and an implementation plan for the use of high throughput sequencing as a diagnostics tool. And so our objective was to develop that strategic roadmap to progress the development and adoption of HGS as a diagnostic tool. And so we established a working group of subject matter experts, and this group, we've been meeting for every 2 weeks for this year, and we're making great progress, and we should have our document to QDAF and Malcolm in middle of October for the next NBC meeting. But you can see from the working group members that we've really tried to get both plant and animal technical experts involved, and from different jurisdictions and organisations. So from across the state, we've got CSIRO involved, but also entomologists and bacteriologists, as well as animal virologists. And this is important because we're always just dealing with DNA and RNA, but we need to have that context of what it all means. And just to give a very high level snapshot of where this framework and implementation plan is heading, we've broken it down into 4 themes, and one is around policy and reporting, where we will generate guidelines and standards required for harmonising HGS usage, data analysis, reporting and data storage. We see a major opportunity and challenge for HGS to be used nationally and effectively is to have a platform. So we want to establish the National Biosecurity Data Platform to bring together sequence information and metadata to allow real-time analysis of disease outbreaks. And the example is AusTracker, like we use for COVID. And that is a really important recommendation we hope that can be adopted, and will take significant collaboration at a national level, but the benefits, as we see daily with COVID pandemic, the benefits will be there. We obviously need to, at a technical level, produce technical laboratory standards for generation and analysis of HGS outputs, exactly like we do for serological assays and PCR assays in our labs. We need to have accreditation around the methods we use, and we need to have our staff proficient in using those methods. So that's a really important part. And at a skills level, the training challenge is quite enormous as well. So we are proposing to establish a national biosecurity network for high throughput sequencing that will deliver training programs and workshops, particularly in bioinformatics. A lot of us are trained as entomologists or biologists and not bioinformaticists. So there's a massive effort to train, or if we train bioinformaticists, to train to align and learn about our ology so that there's a lot of synergy. We want to champion the use of HGS in state laboratories, and we want to promote proficiency testing between labs to get to that degree of consistent standard of applying HGS. So that's what we'll be recommending to MBS, and if it gets endorsed at the next meeting, we really hope we can be active next year and work nationally and collaboratively to get some programs in place to deliver this standard for HGS. I'd like to leave it there, and but happy to take questions in the chat and I'll answer as best I can. Thank you.